about prostaglandins. So even though prostaglandins can produce many unwanted effects such as inflammation, they are also responsible for many beneficial effects. In order to harness those beneficial effects, scientists develop prostaglandin analogs which simply mimic our endogenous prostaglandins. Now, prostaglandins exert their effects by interacting with specific G-protein coupled prostaglandin receptor, of which there are at least 9 known subtypes. The effects of particular prostaglandin may vary widely depending on the tissue and expressed receptors. So now let's discuss some of the commonly used prostaglandin analogs, starting with analogs of prostaglandin E1. Examples of agents that belong to this group are alprostadil, lubiprostone, and misoprostol. Although all these agents are derived from prostaglandin E1, they were designed for different therapeutic purposes. So alprostadil has 2 main therapeutic uses. First is erectile dysfunction. When alprostadil is applied into the urethra, it acts via direct stimulation of cAMP pathway to decrease intracellular calcium levels, allowing for relaxation of trabecular smooth muscle and dilation of cavernosal arteries. This ultimately leads to improved erectile function. Secondly, alprostadil is used in neonates with congenital heart defects to temporarily maintain the patency of ductus arteriosus. So for those of you who need a refresher, ductus arteriosus is a blood vessel found in babies before birth that allows blood to bypass the pathway to the lungs. Although this blood vessel typically closes shortly after birth, keeping it open in certain babies with heart defects may improve blood flow and oxygenation. So infusion of alprostadil relaxes the ductus arteriosus and supports its patency until surgery can be performed. Now let's move on to the next prostaglandin E1 analog, that is lubiprostone. So lubiprostone is used in the treatment of chronic constipation. It works by activating type 2 chloride channels in epithelial cells lining the intestine. By stimulating these channels, lubiprostone promotes secretion of chloride, followed by passive secretion of sodium and water, which increase the liquidity of the intestinal contents. This secretion also stimulates intestinal smooth muscle contractions, which facilitate the passage of stool. Now let's move on to the next prostaglandin E1 analog, that is misoprostol. So misoprostol is another prostaglandin E1 analog, and it is used to treat and prevent stomach ulcers, particularly in patients taking NSAIDs. It can also be used to induce labor. Misoprostol works by binding to the prostaglandin receptor on the gastric parietal cell and causing decrease in intracellular cAMP, leading to decreased activity of proton pump and thus modest inhibition of acid secretion. Furthermore, misoprostol protects the stomach lining by increasing bicarbonate and mucus production. Lastly, by interacting with prostaglandin receptors in the uterus, misoprostol causes softening of cervix and uterine contractions, leading to the expulsion of the uterine contents. Now let's move on to prostaglandin F2 alpha analogs. Examples of agents that belong to this group are bimatoprost, latanoprost, and travoprost. These agents are used ophthalmically for treatment of open-angle glaucoma. They work by increasing the outflow of aqueous fluid from the eye and thus lowering intraocular pressure. Although latanoprost and travoprost accomplish that by interacting with prostaglandin F receptors located throughout the eye, bimatoprost is thought to have a different mechanism of action, which is currently unknown. Furthermore, among the unique side effects of bimatoprost is elongation and darkening of the eyelashes, which makes bimatoprost useful in treatment of eyelash hypotrichosis. Now let's move on to the last group of analogs, that is prostacyclin analogs. Examples of agents that belong to this group are iloprost and treprostinil. These agents are used to treat pulmonary arterial hypertension. They work by increasing production of cAMP, which leads to decreased levels of intracellular calcium in pulmonary vascular smooth muscle cells, ultimately causing vasodilation. This results in significant reduction in pulmonary vascular resistance and enhanced cardiac index.